So, welcome to this class on Neuroscience of Human Movement. So, we will continue with our discussion on action potential. So, this is part 2 of our discussion on action potential. In this class, we will discuss the special case of the voltage gated sodium channel and how action potential is generated, and we will discuss the ionic basis of action potential and how conductance changes during action potential, and we will discuss refractory periods, and we will discuss important characteristics of action potential. We discuss the case where there is a stimulus that causes slight depolarization of the membrane, or when there is a stimulus whose strength is small, then the response is going to be small, and if the stimulus strength is larger, then the response is also going to be larger. We saw this earlier. This we called as graded potential. This is usually caused, the graded potentials are usually caused by what are called as stimulus sensitive sodium channels. Right. In addition, you have what are called as voltage gated sodium channels. These channels are depolarization sensitive. They depend on the membrane potential for their opening and closure. Depending on the value of the membrane potential, they will either remain open or closed. Right. There is a particular point at which this voltage gated sodium channel will open. This point is called as threshold. Essentially, this is the threshold at which this channel is going to open, but in general, when we say threshold is reached, action potential happens. In general, we use this as if threshold is reached for the entire membrane. What is actually happening, threshold is reached for this particular voltage gated channel. It turns out that there are two gates that this particular channel has. These are the activation gate and the inactivation gate. Okay. So, there are two gates that each voltage gated sodium channel has. One is the activation gate and the other is the inactivation gate. In general, at rest, the activation gate remains closed like that, and the inactivation gate is open like that. Right. In general, the activation gate is closed and the inactivation gate is open. Suppose threshold is reached at some point, the threshold is reached, what happens is that the activation gate and the inactivation gate both receive command to change from their rest state. So, let us remember what is the original configuration at rest. At rest, the activation gate is closed and the inactivation gate is open. Both of them must change from their current configuration when the threshold is reached. When the threshold is reached, that means the activation gate will open like this and the inactivation gate will close. However, it turns out that the activation gate opening, that process is a relatively rapid process, is a very fast process. As soon as the command is reached, the activation gate immediately opens, quite immediately, whereas closure of the inactivation gate is a relatively slow process. So, that means there is going to be a relatively brief period during which both the activation gate and the inactivation gate remain open. The activation gate immediately opens and the inactivation gate is trying to close, but not completely closed. In that state, the channel will remain open for a brief period of time. For a relatively brief amount of time, the channel will remain open. Note, this is the extracellular matrix and this is the intracellular fluid. Since a lot of sodium is present outside the cell, a lot of sodium will diffuse through the channel to the inside of the cell. Right. So, this is the activated state of the voltage gated sodium channel, during which the activation gate is open and the inactivation gate is also open, but is in the process of closing. Right. After some time, what happens? The inactivation gate completely closes, like it happens here. In this state, sodium can no longer come inside, this cannot happen. So, that means there is going to be no more sodium entering inside the cell. Now, what would happen essentially for the membrane potential in this case? At this state, this is time in say milliseconds, this is membrane potential in millivolts. So, that is rest state, right, that is rest state. At that, say for example, some small stimulus is taking it to threshold, let us say that is the threshold for example. That threshold will open that gate and close that gate, but this opening is a fast process, this closure is a slow process, we saw that. Because of that reason, there is going to be a brief amount of time during which this is going to remain activated or open. During that time, a lot of sodium will come and take the membrane potential to a very positive value. Note, this is about say minus 60 millivolts, this is about say minus 50, for example, some numbers, typical numbers, right. So, it is possible for the membrane potential to actually cross 0 and become positive, because this channel is going to allow a lot of sodium to enter inside during that brief period. But at around after some time, this inactivation gate is closed and no more sodium is going to enter. At that time, right, that potential is reached, and after some time the potential will actually come down. Why does it come down is something that we will have to see in the future slides, right. So, this is the mechanism by which the voltage gated sodium channel functions and depolarizes the membrane. Okay. So, when the threshold is reached, for example, here there is going to be a rapid depolarization, and this is called as the action potential or the rising part of the action potential, right. But after some time, the membrane potential quickly comes down, actually going way below the resting membrane potential. This is the resting membrane potential, actually it goes way below the resting membrane potential and then comes back to rest, right. It is clear why depolarization is caused. Why is depolarization is caused in action potential? That is because of the action of sodium channel or the voltage gated sodium channel, we saw that earlier, right. Why is repolarization caused? We said at this point what happens? At approximately that point, the inactivation gate closes, that means that no more sodium can enter inside, right. That means it is possible for the membrane potential to remain at that value, but that is not happening, it is not remaining at that value, it is coming down like that, something else must happen. So, what is that something else? That something else is at around that potential, what happens is voltage gated potassium channels open. Now, voltage gated potassium channels, if it opens, we know what could happen, because potassium is present in relatively large quantities inside the cell when compared with the outside the cell, right. So, when a potassium channel opens, a lot of potassium is going to leave the cell, that means the cell will become less positive or more negative, essentially bringing the cell back to resting membrane potential. Actually, this voltage gated potassium channel remains open for a relatively long period when compared with the voltage gated sodium channel, which is remaining open for a relatively brief period, say for example that period, right. In comparison, voltage gated potassium channel remains open for a long period, that it takes the membrane potential below the resting membrane potential, right, like that. So, this is what is called as action potential. What causes this? One more time, another way of viewing the same thing, right. So, the plot in blue here is action potential. So, that is action potential, this is the action potential plot. What you see in red and green here are the conductances of the voltage gated sodium and voltage gated potassium channels respectively. So, that is the red one is the voltage gated sodium and the green one is the voltage gated potassium channel. Now, what I mentioned earlier, the sodium channel and the sodium conductance remains high only for a brief period of time and immediately comes down, whereas the potassium channel, its conductance increases and remains elevated for a relatively long period of time. Note the amount of time for which the potassium conductance remains high, right. Membrane potential goes below the resting membrane potential. Note the resting membrane potential in this case is around that value, is it not? Now, if I erase all this, so that is the resting membrane potential approximately. So, the higher conductance in the voltage gated potassium channel causes this hyperpolarization. Okay. So, note this means that depending on the changes in conductance of the voltage gated channels, you can describe the action potential curve itself. So, this is the peak of the conductance curve for the voltage gated sodium channel, and the peak of the action potential is approximately at the same time, right. And as soon as the conductance starts to fall off, the action potential curve also starts to fall off. And note, actually the falling period of the note that the fall of the potential from the peak is due to multiple factors, due to the increase of the potassium conductance and the decrease of the sodium conductance. A combination of factors come into the picture. So, what happens is that at rest the voltage gated sodium channel is at rest state, basically the activation gate is closed and the inactivation gate is open like this, right. As soon as threshold is reached, the activation gate is open, leading to a lot of sodium entering the cell, right. But slowly the inactivation gate is trying to close. At around that point, that happens at around that point, right. The inactivation gate is closed. After this time, after some time, the voltage gated sodium channel goes back to rest state. This is the rest state, right. But when does the voltage gated potassium channel open? That is approximately at that point, right, for example. So, that is approximately the time when the voltage gated potassium channel opens, and it remains open for a relatively long period of time like that. After some time, it goes back to its rest state. Okay. So, a combination of these factors cause action potential, right. So, some terms one more time, looking at the same thing in one other way and defining some terminology one more time. So, at this stage the sodium channels are open, the potassium channels are closed and a lot of sodium flows in, that means the membrane is depolarized. At this phase the sodium channels are closed and the potassium channels are open and potassium flows out, that means that the membrane is repolarized. This is depolarized with a D, this is repolarized with an R, is it not? There is a period during which you cannot cause one more action potential. Why? At around this time the inactivation gate of the voltage gated sodium channel is closed. So, whatever you do, you cannot activate it. During that period you cannot cause one more action potential by raising the membrane potential. That period is called as absolute refractory period. But at around that point, say for example that point, what happens is that the voltage gated sodium channel goes back to rest state. What is the rest state? The rest state is that state at which the activation gate is closed and the inactivation gate is open. When the voltage gated channel is at rest state, it is possible to cause one more action potential, but already let us compare the potential at this point when compared with the resting membrane potential. Because of extended potassium conductance, this region is at a repolarized state or a hyperpolarized state, right. So, because of this reason the membrane potential is below the resting membrane potential, but the threshold does not change according to that. So, the threshold remains here. Earlier it was sufficient for me to produce that much stimulus to cause an action potential. Now, if you are here, for example, if you are at that point, you will have to produce a much larger stimulus to cause an action potential. At this stage an action potential can be caused, but it requires a larger amplitude. The period during which a larger stimulus can cause an action potential is called as relative refractory period. You can cause an action potential, but you need a larger stimulus than usual, right. So, that is called as relative refractory period. So, this is due to potassium overshoot, this is due to extended potassium conductance, right. After some time, restorative mechanisms bring back the membrane potential back to the rest state or the resting membrane potential. Once at around that point, you are back at rest. So, one more cycle of action potential can be orchestrated at around that point. You can actually start an action potential from here, but you need a larger amplitude. Okay. So, once again showing, so that means this is caused by sodium conductance, potassium conductance, and the action potential is basically a combination of these. Actually, there are other factors, not just these, there are other factors. That is the reason by combining these you will only get a vague action potential, not the actual action potential. There are other factors that are not included in this discussion. Okay. So, changes in conductance of the voltage gated sodium and potassium channels dictate to a large extent the changes in the action potential curve. So, we already saw this. So, depolarization is the process of making the membrane potential less negative, repolarization is the process of bringing back the membrane potential from more positive value. Hyperpolarization is making the membrane potential more negative, say when compared with the resting membrane potential. Flow of positive ions into the cell is called as an inward current, and flow of positive charge out of the cell is called as an outward current. Overshoot is this period when the action potential remains positive, and undershoot is this period during which the action potential is more negative. And we also defined absolute and relative refractory period. Absolute refractory period is this period during which you cannot cause an action potential no matter the strength of the stimulus, because the inactivation gate of the voltage gated sodium channel is closed. Relative refractory period is this period during which you can cause an action potential, but with a higher amplitude stimulus. Okay. This is because the voltage gated potassium channels have taken the membrane potential to a more negative value when compared with the resting membrane potential. This is the reason why you need a higher amplitude stimulus for causing an action potential. Okay. Importantly, the following are to be noted. Turns out that all action potentials are same for a given cell, and an action potential at one site has the ability to cause depolarization at neighboring sites. This is the crucial characteristic of action potential. There is just a lot of sodium entering inside, that diffusion of this sodium causes the remaining areas or the neighboring areas to be depolarized, or the neighboring parts of the membrane to be depolarized, and the neighboring voltage gated sodium channels are reaching threshold, so that they also open and a lot of sodium enters inside. So, action potential at one site causes depolarization at neighboring sites. And note, action potential is an all or none response. Either you have an action potential or you have no action potential. So, it is not possible for you to have a half action potential. Once you cross the threshold, you have a full action potential. In that sense, what you are having is an ungraded response. So, this is binary, either you have one action potential or you have zero action potential. There is no such thing as gradation depending on the strength of the stimulus. As long as the stimulus strength is above the threshold, it will cause an action potential. For whatever stimulus strength, the action potential characteristic will be the same, right. So, it is an ungraded stereotypical response. Okay. So, action potential is an ungraded stereotypical response. And note, action potential always causes depolarization. Compare this with graded potential. We said earlier that graded potentials can also cause hyperpolarization, and note, two graded potentials can sum to produce a total potential change, whereas two action potentials cannot sum. Once the threshold is reached or crossed, one action potential will be caused, and in the meanwhile, if you increase the stimulus strength, it is not going to cause any change to the action potential characteristic. Why? Because you are at the refractory period. So, it will, and also it will not cause hyperpolarization. Only graded potentials will cause hyperpolarization. Action potentials will not cause an hyperpolarization, it will only cause a depolarization. The hyperpolarization period of the action potential curve exists, but you cannot selectively cause positivity or negativity depending on there is no such thing as, or in other words there is no such thing as negative action potential. Action potentials are always depolarizing, right. The amplitude of the stimulus, how is it encoded is the question, right. It turns out that the amplitude of the stimulus is encoded in the frequency of occurrence of action potential. How? We said earlier that this is the relative refractory period, right. Suppose the stimulus strength is relatively high, that can cause an action potential as soon as the relative refractory period is reached, because stimulus strength is higher, then it will cause one more action potential here. So, now instead of it going so much distance back, so much time, taking so much time to go to resting membrane potential and then have one more action potential, if the stimulus strength is high, there will be two action potentials relatively closer to each other in time. So, the number of pulses or the impulses per second will be higher for a higher stimulus, higher strength stimulus. It will be lower for a lower strength stimulus. So, in graded potentials we saw that the response amplitude is what, in graded potentials we saw depending on the stimulus amplitude the response amplitude changes. In action potentials the response frequency or the number of pulses per second changes in response to stimulus amplitude change. Okay. So, the amplitude of the stimulus is encoded in the frequency of occurrence of action potential. Several local potentials or graded potentials can sum and cross the threshold and produce an action potential, but two action potentials cannot add with each other. And action potential amplitude does not reduce during propagation. Importantly, graded potentials start at one point and in the neighboring sites its amplitude slowly reduces. Action potential amplitude does not reduce as it propagates. The reason is because you have strategically placed voltage gated sodium channel on the membrane such that action potential in one of the voltage gated sodium channel will cause an action potential in the neighboring voltage gated sodium channel. Because of this reason the amplitude of the action potential does not reduce. So, for their contribution to the discovery concerning ionic mechanisms involved in excitation and inhibition, the peripheral central portions of the nerve cell membrane, basically describing action potentials and the ionic basis of action potential, Hodgkin and Huxley and Eccles received the Nobel Prize in Physiology and Medicine in 1963, right. So, this has come to be known famously as the Hodgkin-Huxley model of this. So, much of what was discussed in today's class was from the original Hodgkin-Huxley model, except that it is a very simplified version, right. So, in summary what we saw today was the action of voltage gated sodium channel and the generation of action potential, and the ionic basis of action potential, and how the sodium and potassium conductance dictates the action potential voltage and refractory periods, and we discussed important characteristics of action potential. So, with this we come to the end of this lecture. We will continue the discussion in future class. Thank you very much.